Hey everybody, Kelton here to talk to you about biology today. Today we're going to be discussing protein structures, so go ahead, pause the video, take a look at the outline, and we're going to get started. To start off, we're going to talk about a proteome. So you may recall that a genome is basically all of the DNA within a cell. A proteome is basically just all the proteins within a cell. It's possible for two different cells in the same organism to have different proteomes. How do you think this is possible? And great, so hopefully you remembered that proteins are coded for by genes. Genes are just sections of the DNA. So depending on which sections of the DNA or which genes are active, that's going to determine what proteins are expressed within a cell. The basic subunits of proteins are amino acids. Amino acids will combine together to form polypeptides, and polypeptides will make up a protein. All amino acids have the same general structure. They have an alpha carbon, which is the central carbon here. Attached to that, we have a hydrogen, we have an amino group, and we have a carboxyl group here. Along with that, we also have an R group attached. This R group is a variable group, meaning that multiple different things could make up this R group. The identity of this R group is going to determine the identity of the amino acid. So how many different amino acids are there? And great, so hopefully you remember that there are 20 different amino acids. Another question I have then is, why are 1 and 2 structures different? And great, so hopefully you remember that a carboxylic acid can act as an acid at certain pHs and end up giving up this proton here. An amino group can act as a base then, and it's able to pick up an extra hydrogen as we see here. This is going to be the predominant form of an amino acid at physiological pH, which is 7.4. When two amino acids come together to form a dipeptide, a peptide bond is formed as we see here. This peptide bond is just a special type of amide bond. When looking at the figure on the right, we see two amino acids come together to form a dipeptide. What type of reaction do you think is going to be responsible for this? When examining this reaction, hopefully you recognize that this alcohol and this hydrogen are going to end up leaving. When they leave, they form water as a byproduct. Bringing two things together and having water as a byproduct means this is going to be a dehydration reaction. This reaction is going to be assisted by enzymes known as peptidyl transferases, and we'll talk about different aminoacyl transferase enzymes later on. As we see by this image on the right, peptide bonds can be broken down. What type of reaction do you think is taking place in this image? And perfect, so hopefully in examining this reaction, you see 1, something is getting broken apart, and 2, we're using water to break something apart. This is then going to be a hydrolysis reaction. Once two amino acids have come together and we've formed a dipeptide, this dipeptide is going to have specific ends. On one end, we have a C terminus, which is our carboxyl terminus that has a carboxylic acid functional group, and on the other end, we have our amino terminus. This directionality is going to be applicable to all polypeptides, with all polypeptides possessing a C terminus and an N terminus on the opposite side. So now that we've talked about polypeptides, let's move on and talk about the 4 levels of protein structure, which are primary, secondary, tertiary, and quaternary. The primary structure of a protein is simply just going to be the sequence of amino acids. This image does a really good job in showing that the primary structure of a protein is just the sequence at which the amino acids are ordered. So knowing that primary structure is just the order of the amino acids, what's going to determine the order of the amino acids? And nice, so hopefully you remember that genes within our DNA are going to code for the order of these amino acids. Secondary protein structure is just local folds of the polypeptide chain. This is going to be determined by hydrogen bonding on the protein backbone. So as a quick refresher, do you remember what's required for a hydrogen bond to take place? Remember, for hydrogen bonding to take place, you need a hydrogen that's bonded to an oxygen, a fluorine, or a nitrogen. So knowing the requirements for hydrogen bonding, what parts of the polypeptide backbone then do you think are going to do hydrogen bonding that results in a secondary protein structure? And nice, so as you can see in this image, our hydrogen bonding at the polypeptide backbone is going to take place between our amino and our carbonyl groups, as highlighted in red. Two really common elements to secondary protein structure are our alpha helices, as we see here, and our beta pleated sheets, as we see here. Tertiary protein structure is a little bit more complicated than secondary protein structure because now we're talking about the whole 3D shape of our polypeptide. This is going to be caused by interactions between the R groups on those amino acids. Can you recall any of these R group interactions that may influence a polypeptide shape? And great, so there's quite a few different R group interactions that can influence a protein's shape. A few examples are ionic bonding, as we see here, hydrophobic interactions, like we see there, and hydrogen bonding, which we see here. Hydrophobic interactions are relatively important because we see this is going to influence a protein shape by putting any hydrophobic R groups, as we see here, to the inside of our polypeptide chain. Another important component of tertiary protein structures are disulfide bridges. Disulfide bridges can be seen here, and they exist between two cysteine amino acids. These are covalent linkages that are really strong, and they help act as staples to hold that protein in its most stable conformation. The last level of protein structure is quaternary protein structure. Quaternary protein structure involves large proteins with multiple polypeptide units. Why might it be important for us to take into consideration the structures and the organization of each of these polypeptide subunits? So why this is important is because a protein's structure is directly related to its function. For example, if we look at hemoglobin, which is a tetramer protein, which means it has 4 subunits, we see that its function of being able to carry gases throughout the body is only possible due to its structure and its shape. Lastly, we are going to talk about denaturing proteins and what that does to the shape as well as the function of a protein. So take a second and use this image on the right to see if you can decipher what denaturing a protein might mean. Hopefully in examining this image, you notice that these proteins on the left are all very curly. Once they've been denatured, they're all really straight now. So we lose those higher order elements of protein structure. But notice the primary protein structure is still retained in most cases of denaturation. So what does a loss of shape cause for a protein? And great, so hopefully remember the example of hemoglobin where its function is really specific to its shape. If a protein loses its shape, it's also going to lose its function. I've already given you a freebie over here that heat is something that we use to denature proteins, but can you think of any other examples or things that might denature a protein? And great, so temperature and pH changes, chemicals, and radiation are all things that could possibly cause a protein to lose its structure. When some proteins are denatured, they're able to refold again. Other proteins cannot do this though. So if we take a look at this egg, what is a protein in that egg that might not be able to refold? So ovalbumin is a protein in an egg that can't go back to its folded state after being denatured. If we think about cooking an egg, we started off with a lot of clear, runny stuff, but now after cooking the egg, it's all turned white and it's almost very rigid. So no matter what we do to this egg, we are never going to be able to get this white to go back to that clear and runny. A protein's shape is going to be key to its function. So as you might have guessed by now, there's a lot of different functions of proteins. You can see a table of different protein functions here, and we're going to break these down further throughout your course. So now that we've finished with the lesson, it's time for a mini quiz. So, which of the following does not involve hydrogen bonding? Primary protein structure was just the sequence of amino acids, so I don't think that involves hydrogen bonding, so let's keep that one around for now. Secondary protein structure was caused by hydrogen bonding among carbonyl and amino groups on the polypeptide backbone, so we can get rid of this one. Tertiary protein structure was influenced by R group interactions, and one of those interactions was hydrogen bonding, so let's get rid of this one too. Beta-pleated sheets, if you remember, are just a form of secondary protein structure, so we can get rid of this one as well. And great, so this leaves us with A. Primary protein structure does not involve hydrogen bonding. Okay, great, so now that you've had your lesson and your quiz, let's go ahead and do a review to see what you learned today. So first, what type of intermolecular force leads to the formation of secondary structures in proteins? Next, in an aqueous solution, hydrophobic interactions cause a protein to fold in a way that positions the nonpolar R groups where? And lastly, why do proteins lose their function when denatured? If you can answer these questions, you're well on your way to success. Everybody be sure to have a great day, and I will see you in the next one.